Hello everyone, welcome to MDC Connects 2021. I'm Sarah Brockbank, I'm a lead scientist at the Medicines Discovery Catapult, and I'm going to host the webinar today with my colleague Darren Holmes. MDC Connects is a series of weekly webinars that runs on a Tuesday at 2 o'clock. So MDC Connects this year is focusing on complex medicines, and we're joined by a host of experts from across the community that are going to help us go from concept through to the clinic. And we're here on the third in the series today, and I appreciate that most of you, I think, are joining us every week. But for those of you that haven't heard the first two seminars, and indeed, well, you won't know if you're not hearing now, but for those that haven't heard the first two seminars in the series, these are available as recordings on the MDC website. But for today, in this session, we will cover the analyses available to understand our complex medicine and how it's behaving in the body, in tissues and in cells. And I'm going to welcome three speakers. Firstly, Zara Rattray from Strathclyde University. Zara's going to talk about the fate of complex medicines in the body, the role of the protein corona and how we measure it. Then Jane Lawrence from Manchester University is going to take us intracellular. She's going to demonstrate some model systems which can be used to measure and optimize nucleic acid delivery to the cell. And finally, Rob Weller from LGC is going to tackle the subject of bioanalysis of complex medicines in order to understand PK and exposure of the drug in the body. We will take questions at the end of each of the presentations, and if you wish to ask a question, can you please put it in the Q&A box at the bottom of your screen. Please we're going to address them at the end of each of the talks, so if you can put your questions in during the talk so they're ready for me to ask them when the speaker has finished. So without further ado, over to you, Zara.
Good afternoon, everyone, and thank you very much, Sarah, for the introduction, and also the Medicines Discovery Catapult for the invitation to speak here today. So I hope that you can all see my slides. Can I have a thumbs up, please? Yeah, awesome. Thanks, Sarah. So my presentation today is going to look at the early assessment of complex nanomedicines and prototypes, and trying to characterize the interactions that occur between a nanomedicine and biological systems. So to give a bit of an overview as to what I'm going to be talking about today, if we imagine a nanomedicine and a nanomedicine prototype, what we're looking at essentially is a very highly reactive particle that's going to interact with its surrounding environment. Usually quite a lot of investment goes into the early characterization of these prototypes from a critical quality attributes perspective, so looking at particle size, surface characteristics and properties. But there is an added layer of complexity and another element, which is looking at interactions with biological systems and how these take place. So, for example, if we dose a nanomedicine via the intravenous route of administration, we will see that nanomedicine encounters and experiences blood flow. It will interact with surrounding blood cells and biomolecules within the circulatory system. And these interactions will then subsequently lead to surface adsorption of those components onto the nanomedicine, essentially conferring a new biological and chemical identity. So this biological and chemical identity changes as a function of the critical quality attributes of that product, which could be the size, surface charge, characteristics or composition. So again, this is very unique to the particle type, and even the payload may well have an impact on how the protein corona is formed and how these interactions take place. And then this is also a function of the environmental parameters that the particle will encounter and experience, such as the media in which it is dispersed, the composition of that media, and also physical parameters relating to flow rate. So the proteins that adsorb onto the surface part of a nanoparticle or a nanomedicine are known as the protein corona, a term that was originally coined by Dawson and co-workers. And the way the protein corona is perceived is to be a very dynamic structure. So usually when we talk about the protein corona and most measurements are performed, we're referring to the hard corona, which is the proteins that are bound to the surface of the particle. And also the soft corona, which is loosely associated and bound to the particle surface. So having given a little bit of an overview and a whistle-stop tour on what the protein corona is, why should we care and why should we perform measurements of the protein corona? So the protein corona has a fundamental impact on the biological and chemical properties of a particle. And from what we understand in the literature in terms of in vivo findings and preclinical studies, is that it can have an impact on interactions with cells. So this can have an impact on interaction with immune cells and their subsequent activation. This could then have a knock-on impact on circulation times and organ biodistribution. Also, the colloidal stability of the particles changes as a function of the protein adsorption, especially where physicochemical parameters change when proteins bind to the particle surface. And then this also goes on to have a knock-on impact on drug release characteristics. And also, if we're looking at whether these particles become more sticky and start to agglomerate together, then what we might start to see is the formation of depots. And as you will all be aware, drug release from a depot versus a single particle is a very different model. So hopefully I've convinced you that there are many different reasons for why we want to actually measure the protein corona, and also the fact that this might not necessarily be a very simple and straightforward process, just through the simple fact of looking at how many different processes to do with the product performance are impacted by the presence of the protein corona. So when we look at the biological fate of a nanomedicine or a complex medicine and how it interacts with the biological environment surrounding it, we can look at multiple tools and models to perform these analyses. So today I'm going to mostly be looking at the bioanalytical tools and methodologies that are routinely implemented in the protein corona, and then hopefully have a line of sight to what the future may look like in the context of analysis of the protein corona for complex medicines. But just to briefly highlight, there is a very diverse wealth of in silico tools that are available out there. First of all, to look at how the particles are dispersed in the circulatory system upon injection. And then also looking at interactions between particles and proteins. So using atomistic modeling or molecular dynamics modeling, we can predict some of these interactions and how they're going to take place. And there are more and more models that start to take this into cells and tissues and predict cellular interactions, how various different intracellular trafficking pathways and uptake pathways may be impacted. And this actually is something that Jane Lawrence is going to cover in the next talk as well, looking at cellular models. And then we can look at this in a more systemic way, so looking in preclinical models and humans as to what the biological fate is. And each one of these tools provides a different opportunity and a different step in which we can either characterize the protein corona or predict what impact it's going to have on product performance. And Rob, who also joins us here today, will be talking a little bit more about the cell tissue human element and the bioanalytical complexities of looking at the biological performance of complex medicines. But today I'm going to, as I mentioned before, focus on the bioanalytical piece and the various tools that are available in the characterization of the protein corona. So the majority of studies that have been performed to date in this space and area have been predominantly around looking at metallic and inorganic delivery systems. A typical pipeline for measuring the protein corona and its analysis is included on this slide. So the most common and typical approach is to treat a nanoparticle within either a protein containing media or blood products, so looking at plasma, sera, or even cell culture media to look at how the particle will interact. There will normally be a period of co-incubation. Mostly this takes place under static conditions based on the most majority of reports that are available within the literature, and it will normally take place at physiological temperature. Then there's an element of recovery and isolation of those particles. So quite often these techniques rely on some separation step to remove the particles from the bulk solution in order to ID the proteins and look at the impact of the proteins on particle parameters. However, in recent years, there's been more and more emphasis on using more in situ techniques, and techniques that are quite gentle so that they're minimally disruptive to the protein corona and its composition. And then downstream of that, what is routinely and commonly done is fingerprinting and qualitatively looking at changes in that fingerprint of proteins that have adsorbed onto the particle surface. So SDS page is normally used. Sometimes the bands that are separated through SDS page will be digested, trypsinized, and put through LCMS for proteomics-based fingerprinting. And in parallel, quite often, some groups have also measured the impact on physical parameters. So using particle metrology tools to look at changes in particle size and concentration, which can give us an indication of the colloidal stability changes that occur as a result of protein corona formation. What I've also included on the right-hand side panel of the slide is some other elements of the protein corona that we might want to know more about. So looking at the conformational analysis of the surface adsorbed proteins, as it has been known to have an impact on how it interacts with various different receptors and biological targets. So some of these tools rely on vibrational spectroscopy and NMR to look at protein conformation for the surface adsorbed proteins. We might want to look at total protein quantification of how much protein in total has been adsorbed onto the particle surface. And that's where proteomics, protein quantification assays such as the BCA and Bradford assay are useful. And in some cases, people also measure the thickness of the protein corona, and that's where we have increasingly seen the use of cryo-TEM. So for those interested, actually, there was a talk last week through the same MDC Connects webinar series, which looked at the application of cryo-TEM in complex medicines. So I highly encourage you to visit that if that's something of interest. Also, in terms of IDing proteins, we can use mass spectrometry. And in some cases where access to mass spectrometry might be prohibitive for some, SDS page coupled with Western blotting may be an alternative approach if we know what the most commonly occurring proteins are going to be and the most abundant proteins adsorbing. And then we've also got the question of ELISA assays as well, where you can perform some quantification. In addition to that, at that sort of prototypic stage of design, we may be looking at affinity of interaction with proteins and dynamics for that, where we can look at isothermal calorimetry, or using the quartz crystal microbalance approach to look at dynamics of interactions, affinity and rate, which all of these can provide very useful information in terms of how fast these processes are taking place, what the identity is, and we can optimize this for each particle prototype. A word of warning, no single approach on its own works, as you would have learned through complex medicines. Quite often we have to rely on orthogonal methods, and usually more than one approach may be used to perform these analyses. And there is definitely a drive to move more towards the in situ based analytical approaches. So what I'm going to cover next is an example of some of the work that we have been doing within my laboratory, and my co-workers have been really busy with this, which is trying to develop more sort of in situ approaches for that physical analysis. So can we actually perform the treatments? And these treatments were all carried out under end-over-end rotation at physiological temperature in conditions mimicking cell culture. So we're quite interested in that very initial stage of how do these interact, what binds to the surface, and then what impact does this have on the physical parameters, and how will this impact cellular uptake? So is there a change in mechanism of cellular uptake? So some of the preliminary findings we have, so on the left-hand side panel is looking at changes in dry mass using the resonant mass measurement approach. So I've actually put a picture of the analytical techniques. I'm aware that resonant mass measurement may not be something that everyone's familiar with. It's a microfluidics-based approach which measures particle mass. So we can collect particle mass data as well as particle diameter, and this is really useful in the context of being a non-optical method. Also, the limit of detection using the nano sensor channel is something around 100 nanometers. So for these particles that were 100 or 200 nanometers in size, The beauty of it was that you just pick up the signal from the particle or the protein that is bound to the particle surface. And what you can see from this figure is, as a function of time, we see a significant change of mass, especially as we head more towards a 24-hour time frame. And we also see that maybe these interactions are not necessarily the same and homogeneous across the entire particle size distribution. And in fact, it's ranged. So we can see at earlier time points that some of these mass changes result more from protein-based interactions with the particle. And then as we move on, we see more nanoparticle agglomeration occurring in the samples. These findings, we were also managed to corroborate these findings with nanoparticle tracking analysis, which is the two sets of graphs that we have on the right-hand side of the slide. So we were able to reproduce these observations and obtain very similar results for performing that in situ analysis without nanoparticle recovery. And then in the interest of time, moving on to the more sort of emerging approaches that we're seeing more and more in the literature. I've got an example of a study here that used electrical field flow fractionation with online multi-angle light scattering and nanoparticle tracking analysis to characterize changes that occurred in particle size, zeta potential, and the RG and DH, so the hydrodynamic radius and the radius of gyration. And again, this is an inline analysis. It's pretty gentle and it's minimally disruptive compared to using centrifugation wash steps, which can completely change the identity of the particles that bind and interact. And what you can see here is that they were able to perform the comparison as a function of medium. So one thing I wanted to bring everyone's attention to is the fact that at Strathclyde, we're actually investing quite heavily in a metrology facility that will have multiple field flow fractionation modalities. So if this is something that you are interested in, we're always looking for collaborators and users of the technology to speak to. So this will be a multimodal FFF and also it will have orthogonal size exclusion chromatography, and we'll also have online RAM and ICP-MS and light scattering capability. So if this is something you're interested in and it interests you, please feel free to reach out to me after today's webinar series. One thing that I wanted to kind of finish and close this presentation with really is there's a lot of emphasis on the development of analytical methodology, but very little about biologically relevant conditions and mimicking these. If we're looking at intravenous administration, we want to look at blood flow conditions, and what the study really showed is that flow conditions can significantly alter the results we get compared to just simply co-incubating a particle with protein-containing media. So what does this actually mean, and what's the line of sight? So we're in the era of precision medicine. We're wanting to look at using big data. We're wanting to incorporate this and translate this into medicines that are efficacious and have minimal adverse events. So the way I kind of see this going is we can use high throughput microarray microfluidics based analyses mimicking biorelevant flow conditions and even compositional conditions to perform these measurements. We can multiplex these with the latest in separation and analytical capabilities. And then in turn, we can perform some training so that we can actually predict prototypes from a very early onset and optimize these in product design. So I've pretty much covered most of these points in the talk today. I think it's really important in the characterization of complex medicines to apply biorelevant methods and to also look at a combination of both in situ and ex situ approaches so that we can ensure that what we're observing is not an artifact of the technique being used, and to also look at more harmonization in how we perform and report experiments looking at biological interactions, and also ultimately develop an interactome passport for every prototype. And the more that we understand and the more that we build libraries, hopefully this can pave the path for future therapies. So I'd like to thank the MDC again for the invitation to speak today, and thank all my past and present colleagues and also the funding bodies who have enabled us to perform the research that we have done so far. And thanks to everyone attending the webinar today. So I'm more than happy to take questions.
Hi, Zara. Thank you for that talk, very interesting talk. One of the questions that I'd like to ask on this is, what data do we have currently that shows the difference of the composition of the protein corona between your in vitro and your in vivo, and would we expect that you might get a different protein corona from an in vivo animal to the clinic, you know, that translational piece?
Yes, so to basically break this down, there are a few articles where they've looked at preclinical administration of prototypes, and then they've removed samples at various different time points and then performed analyses on the protein corona composition. Not very many studies have actually been done in the space to actually mimic these conditions in vitro. So, for instance, use in vivo mimetic conditions to reproduce what the particle would encounter. So quite often there is a bit of a difference between the in vitro and in vivo protein corona composition. So I think we are still quite a way in terms of trying to develop methods that are biomimetic. And I think there's definitely a lot of scope and potential in that space to kind of reach that point. And I think it's very important to always validate these assays with preclinical studies as well. And I can't stress that enough.
Okay, brilliant. And a second one, this one, where is our understanding regarding what we see with the protein corona and what we want to have with the protein corona so that we can engineer the nanoparticle?
That's a very excellent question, actually. So I think we're still quite away from doing a structure, composition, function for these, and that's something my laboratory is certainly very interested in, but we're kind of at that stage where we're like, let's understand the structure properly and then translate that into a function. So we're actually trying to start to develop capability where we couple proteomics analyses with the physical parameters, and then look at how this on different scales, so looking at how they interact with immune cells, how they interact, for instance, with cancer cells, and then ultimately in tumor-bearing preclinical models, how these would interact in their environment. So there is some understanding, we do have some basic understanding of composition function, but I think we're still quite it's my dream ultimately to be at that point in about 10-20 years time where we can predict what the composition would be based on the parameters and then reverse engineer particles. But I think that's kind of the direction of travel we should be looking to head in and learn from previous experiences.
Brilliant, thank you, Zara. So our next speaker is Jane Lawrence from Manchester.
Okay, I know I'm on. Okay, I can't manage to get myself off. What do I do?
There you go.
Okay, thank you very much. Thank you, Zara, for that. I'm going to be talking about the interaction of colloidal gene delivery vehicles with model membrane. And I'm really using this as an exemplar. So I could be talking about virtually any nanoparticulate system. I could be talking about any any membrane. But today I'm going to be talking about the interaction of gene delivery vehicles or lipopolyplexes with the endosomal membrane. Lipopolyplexes are formed really easily by preparing vesicles from containing a cationic lipid, and you can see on the right hand side we've got the DODAB, which is the cationic lipid I'm talking about today, with or without the presence of the helper lipid DOPE. You make the vesicles, interact with nucleic acid, which is here with cDNA, and you get the lipoplexes formed really easy. As you can see, they're small spherical structures that consist of bilayers of lipid. Between each bilayer is a water layer in which the DNA resides. We routinely characterize these lipopolyplexes, and typically they're of the order of 110 to 120 nanometers. If you look on the right-hand side, you'll see mention made of the preparations being no different in the presence of deuterated DODAB or in D2O. And this is just to say I'm going to be talking a little bit later about neutron reflectivity, in which case we actually make the preparations with various states of deuteration. So this is just to tell you that when we do that, it makes no difference to the properties of the particles. So why are we looking at their interaction with nanoparticles with endosomes? It's obviously something I don't really need to talk to in this audience, but just to say when particulates are taken into cells, either by a phagocytotic process or by mediated endocytosis, they typically go into the endosome, generally known as the early endosome, which then matures into the late endosome and eventually into the lysosome, which is effectively the dustbin of the cell. And if we're going to deliver drug or nucleic acid material into the cell, we really want that material to get out of the endosome, certainly before it gets into the lysosome and ideally before the late endosome. And we've got models of lipids that we picked to actually mimic both the early and the late endosome. So the early endosome has 4 lipids that we used. You can see 2 unsaturated lipids and 2 more saturated lipids, and you can see the compositions of the various lipids. So that's the early endosome, and then we had a model of the late endosome, and in this particular case we don't have any sphingomyelin present, but we've replaced it with another lipid which is slightly more unusual, called LBPA, which is an anionic lipid that is present in the late endosome, and it's got some very interesting properties. You'll see that the amounts of the various lipids slightly alter in the various models. So that's our basic system that we're looking at. And to look at the interaction between the gene delivery vehicles and the endosomes, we use 4 techniques, which I'm going to tell you a little bit more about these techniques. They're all lab-based techniques with the exception of specular neutron reflectivity. This is accessed at a facility. If you are an industry that's interested in using this type of technique and in working with an academic and funding that academic, there's a possibility to gain access to this technique via the facility. If you want to know more about that, I can tell you a little bit more offline. So what we were doing with Langmuir trough experiments, for those of you who don't know it, it literally is a trough, and that trough is hydrophobic. So when you fill it with water, the meniscus of water stands proud to the trough, which means that you can put a known amount of lipids on the surface, measure the surface tension in the presence of lipids, and then by moving barriers that go across the top of the trough, you can actually compress those lipids together and measure how surface tension changes. Why do we want to do this? Well, the monolayer is actually quite a good model for half a cell membrane. And if you compress that monolayer to a surface pressure about 30 millinewtons per meter, it's actually quite a good mimic of the lateral pressure present within a cell. So they're actually quite simple, but quite good models of what's going on in a cell. And what I've just shown you on the right-hand side basically is the surface pressure isotherm that we get by compressing either the early endosomal model or the late endosomal model. Again, I make mention to the fact it's no difference if we've got deuterated materials present. The main differences between the 2 models is that the late stage endosomal model is slightly more expanded. So that means that the lipids are probably in a more fluid state than the early stage membrane. So that's our basic model, and we use that model throughout a number of experiments I'm going to tell you about in a minute. But the first is, it's a trough experiment when we added the lipoplexes to the underside of the monolayer and examined how the lipoplexes interacted with the monolayer. And you can see on the right-hand side, I have some of the data. So if we just take the top left-hand graph, what happens here is we measure surface pressure. As we compress it, we get up to 30 millinewtons, which is, as I said before, the lateral pressure in the membrane. Check it's stable. Once we've got that, we inject, indicated by the arrow, the lipoplexes, and you can see straight away there's some interaction between this particular lipoplex and the membrane. So the top 2 graphs represent the early endosomal membrane, the bottom 2 the late endosomal membrane, and lipoplexes with dope and without dope. So you can see that when we've got lipoplexes with dope, and remember I said this is a lipid that we think alters the integrity of the endosomal membrane, there's a much greater interaction than when we actually don't have that particular lipid present. So we can see differences now between the lipoplexes, and we can see the lipoplexes interacting with the membrane. What we don't know is what materials from the lipoplexes have gone into the endosomal membrane models. We then went on to use a technique called Brewster angle microscopy, which again uses a Langmuir trough setup, But what we do here is shine a laser onto the surface of the monolayer. And if you shine that laser on at an angle of 53 degrees onto a surface of water, you get no reflection from the surface of water, so your picture looks black. If you actually have lipid on the surface, and lipid has a different refractive index than water, when you shine that laser on the surface at the same angle, you get reflection, and you can see a picture of what the surface looks like, which is exactly what we've got in these particular pictures here. So A is just a picture of the endosomal membrane in the absence of any lipoplexes. And just to say that that surface stays the same, looks no difference for 2 hours. So any changes we see in the presence of the lipoplex are actually due to the interaction of the lipoplex with the monolayer. First thing to say is you can see some little white bits there, which probably suggests that the monolayer, even in the absence of lipoplexes, is not homogeneous. When we add the lipoplexes, the top one is in the absence of dope, and here are lipoplexes with dope. You can see an increase in the number of the little white domain regions, and this interaction occurs really quickly within 30 minutes, which is exactly what we saw at the trough. And you can see there's slightly more interaction, or you'll have to believe me, slightly more interaction when we actually have lipoplexes containing dope. So again, we're seeing this differential interaction between the lipoplexes in the presence and the absence of the helper lipid. Just to show that we've looked at the late stage endosomes, and basically we've got similar results. So something that is coming out of these experiments is that we're not seeing much difference between the early and the late stage endosomal monolayers. So at the moment we know that the dope interacts very strongly with these models, but we don't really know how much material is going into this monolayer, and indeed what particular type of material is interacting with these model membranes. And to do this, we actually turn to neutron reflectivity, which basically uses exactly the same Langmuir trough setup. But this time what we do in addition is basically shine a neutron beam at the surface and measure the pattern of the scattering. And by looking at layers and lipoplexes that contain deuterated materials, we can actually highlight the systems that the parts of the system that we're interested in. It's almost like a really expensive microscope. You can see inside the structures that you want to look at. So the data I've got here, it's just the data we get for the early and the late stage endosomal monolayers, and this is in the absence of lipoplexes, and we can get ideas of how thick this monolayer is and how much water is actually present. What we're really interested in is what happens when we add in the lipoplex, what happens to these layers, and this data shows us this, and this is an experimental technique. We can actually quantify the amount of material going into the layers. So basically these black lines are the original data set. And when we've got red lines, this is lipoplexes that contain deuterated material. And this time we could add the deuterated cationic lipid. The cyan line shows the data when we just add the normal lipoplex with no deuterated material. There's clearly differences between the data, although obviously the differences are not particularly great. So these are the early endosomal monolayers, and these are the late endosomal monolayers. But by fitting the data, we could actually say that we know that 10% of that layer after we added the lipoplex, in the case of the early endosomal monolayers, is actually the cationic lipid. Whereas with the late stage endosomal monolayers, we had 15% of the cationic lipid go into the layer. So we actually now quantifying these effects. But what we don't know is whether the dope went into this layer. And the reason why we don't know this is not that the technique doesn't tell us or can't tell us, but in our particular case, we didn't have available to us a deuterated version of dope. If we had that available or could have easily made it, we could have actually had that information, but unfortunately it's not that trivial to make. But you can see with this technique, we can quantify things now. So the final technique I used, and this is not a monolayer technique, this is a membrane technique, is giant vesicles. And this is a very useful visual experiment to see what's going on. Giant vesicles, as you can see by the error bar, these are about 20 microns, 40 microns in diameter. So these are very large and are actually quite fragile. And you make them by depositing lipid on electrodes, and adding solvent, which is water in this case, and water containing dyes, you'll see in a minute, puts electric current across, and we make the vesicles. And then we can examine those vesicles in situ in a confocal microscope. So when we make the vesicles, we make them in the presence of calcine, which, as you know, is green. And what we want to do is to see only the vesicles and not the surrounding solvent. So we add calcium into the solution, and that calcium will complex the calcine. So on the outside, we've now quenched the fluorescence, so we only see the fluorescence inside the actual vesicles. And then what we can do is add lipoplexes and see the changes that occur. So the top row of figures are what happens if you add lipoplexes containing dope in this case. And the dope is actually labeled red because it's rhodamine, and that's what you can see the red color, that's the dope molecules. So we add lipoplexes with dope in this case, and we look at the changes, and this is to the early endosomal membrane, and this is to the late endosomal membrane. And you can see very clearly that the dope is going into the endosome, which ties in with all our other experiments. And with time that increases. But interestingly, what we also see is that as the calcine coming out of, or looks like it's coming out of the actual vesicles, certainly it's becoming less intense over time. So that suggests to us that actually the dope is altering the integrity of the membrane, which again is perhaps not too surprising because this has been proposed before. We could look at this in a little bit more detail by repeating the same experiment, but adding in now Dappy, which is a blue stain that lots of people use to label nucleic acid in cells. We were using it to label free DNA. And what's really interesting here, you can again see the calcine color, the green color disappearing over time. If you look carefully, you can see patches of red, but basically around the vesicles, it looks purple. That's because there's both the rhodamine labeled dope there and the Dappy. And over time, the blue color becomes quite intense inside the vesicle. And that's because we believe that DNA is translocated into the vesicle. So in other words, the dope has affected the integrity of the membrane and allowed the DNA to go inside the vesicle. Now it's quite a whistle-stop tour. Hopefully I won't go to slide in a lot of detail. Hopefully you'll see that different techniques give us different information about the system that we're looking at. And I think that's really important. And certainly Zara alluded to that, the importance of using several techniques together to get a good picture of the system that you're really interested in. But just to summarize, in these studies, we found there was little difference between the two endosomal monolayers that we were looking at. We could see that both DODAB and DOPE entered the endosomal monolayers, and the presence of lipid actually altered the integrity of these membranes. And hopefully I've shown you that the combination of techniques are really powerful in understanding the systems that you're interested in. Just to add, there is another technique that we use at Manchester that I've not spoken about today because we didn't use in this study, is a technique called ellipsometry. And that can also add a little bit more understanding to the system that we're interested in. So that's all I really wanted to say, except I really wanted to thank my co-workers, Alexandra and Layla. And thank you for your attention.
Thank you, Jane. Thanks very much. So I was wondering, are these model systems that you're running at Manchester, are they available for other researchers to screen their molecules?
Yes. We could either supply them or give them how to make them. They're very simple. Yeah.
Okay. And the other thing is that you showed this for a DNA cargo. Is it for any nucleic acid cargo?
Yes, it could be used for any nucleic acid and lots of other delivery systems as well. It doesn't just have to be for DNA. It could be for, I mean, you can do the same thing for LNPs, for example, which you all know are being used as vehicles for some of the vaccines, the COVID vaccines, that you can study exactly the same way with these systems.
Okay, brilliant. Thank you, Jane.
Thank you.
So over to you, Robert.
Okay. Thank you, Sarah, and thank you to the MDC for the invitation to speak today. So what I'm going to be talking about is the bring it back around really, and talking about the importance of measuring pharmacokinetics and bioanalysis within drug development, and some of the challenges that we're likely to face with the bioanalysis of complex medicines. Just a slide to introduce our group Drug Development Solutions at LGC. So we're split across two key areas really. We have our bioanalytical sciences group, so located up in Fordham, Cambridgeshire, and our analytical and material science group, which are co-located at the Fordham facility, but also at the research park in Sandwich, Kent. Okay, so I guess going back to basics really, and you know, just looking at the question of why we want to measure in vivo drug concentrations. And when we talk about measuring in vivo drug concentrations, what we're actually really interested in is measuring drug concentration versus time. So we presented with a profile like this on the right-hand side. And the study of these drug concentrations, we kind of term as pharmacokinetics. So essentially what the body is doing to the drug after it's administered. And understanding the pharmacokinetics of a drug is really important in that we can start to link the drug or excipient concentrations to the toxic effects they're having and also their efficacious effects. So then we can start doing things like making predictions of safe dose levels in humans, designing suitable dose levels and frequencies of dosing within humans as well. You know, we can then kind of take a step back and look at those pharmacokinetic parameters and start to really design desirable candidates to progress through development based on those characteristics. And you know, ultimately what it's about is, you know, making sure that we are achieving this exposure within the therapeutic window of the drug. So we're not dosing so that we're causing toxic effects, but we're dosing enough that we're having that efficacious effect. So that's why measuring pharmacokinetics is important, but actually when it comes to measuring it, how do we go about doing that? And this is when we look to the field of bioanalysis, and there are some really well-established techniques out there in the field for measuring those drug concentrations within biological samples, and you know, they're to regulatory requirements as well in terms of characterizing the assays as well. So you know, they're fairly robust techniques. Typically they can be performed by LCMS or immunoassay. And the drug product is used to construct a calibration line. And then we can measure the unknown sample response against this calibration line and back calculate the concentration of that sample response. You know, generally considering molecular entity that we are measuring to be reflective of the whole drug product. So when we consider the challenges in the bioanalysis of complex medicines, that's where it starts to get a little bit cloudy. And a couple of things that we need to consider, you know, with these complex medicines is the increased heterogeneity that they bring, both in their chemistries and their formulations. And as Zara pointed out in her presentation as well, you know, that complexity can really be increased when those molecules are introduced in vivo. So we need to be asking questions around, can the assay achieve measurement of the entire medicine? And actually, does measurement of the whole medicine have any relevance? And then we need to think about as well, achieving a measurement that is reflective of the state in the biological system. So can we maintain the integrity of that medicine or that formulation through sample collection, through storage, through extraction before the analysis technique? And also thinking about, does the drug product we're using to construct the calibration standards really reflect the drug product that is actually in the sample being measured? And you know, there's a couple of examples there of a drug dendrimer conjugate, which is slowly releasing encapsulated drug over time, potentially. And then a nanoparticle as well, which you know, just through sheer complexity and size, it's going to be really difficult to measure that kind of molecular entity. Okay, so I guess I wanted to present a case study to demonstrate some of the challenges, and the case study I've chosen is an antibody drug conjugate. So ADCs as a therapy have been used in treatment of cancer for a number of years now, and there are actually 10 ADCs that have gained market approval to date. They offer the high specificity of an antibody therapeutic, so they have the ability to bind to tumor-expressed antigens, which then undergo receptor-mediated endocytosis. These antibodies are coupled to a small molecule cytotoxic payload, which when that endosome undergoes lysosomal fusion, we end up with release of that payload either through degradation of the antibody or some kind of cleavage of the linker between the drug and the antibody. Essentially, the end result is the same. We get an interference with critical cellular processes and ultimately leading to apoptosis of that cell. So we can see why there's I guess a drive for ADCs. They can offer a much wider therapeutic window when compared to traditional chemotherapy. So bringing it back round to bioanalysis, and when we consider the challenge that ADCs present, they exist as a fairly heterogeneous mixture, generally as a drug product. So we have a complexity of conjugation sites where the payload can be connected. We have biotransformation as well, which can lead to cleavage of the payload at various sites. And then we can have the payload itself circulating freely or as a metabolite as well. Currently, we don't have a single assay that can measure all these forms, but what we can do is look at subsets of this heterogeneous mix and analyze those subsets. So we can look at total amount of antibody. So that can be the antibody molecule with payload attached to a certain degree, or with no payload at all. We can look at conjugated antibody or conjugated payload. So essentially an antibody with one or more payloads attached to it. Or we can look at the free payload and metabolites of that free payload as well. So I guess the next question is, how do we know what assay to select? And really that comes down to understanding the specific question that is being posed by that project at that particular time to really drive that assay selection. But we also need to consider some limitations around measurements that we can achieve. So considering the molecule, there are different conjugation techniques of the payload. So we can have lysine or cysteine or site-specific conjugation, which can increase or decrease the heterogeneity that we're likely to see. And that can have an impact on the assay selection. The linker technology. So I mentioned before about having a cleavable linker. Some have non-cleavable linkers. And again, that can really drive the selection of assay. And then we have the more common limitations in terms of reagent availability. And, you know, just thinking about raising antibodies against small molecule payloads, that can be a challenge in itself. And then lastly, you need to consider the laboratory technology that they have at their disposal and their expertise as well. So that being said, what I'm going to do is just have a run through of each of these specific assays and show some limitations and what they may actually be used for and results that they may give. So first up, we have the total antibody measurement. I guess it's obvious, but we can't discriminate if the molecule is going to be active. So whether or not it's actually got a payload attached to it. But it can give us an idea of the pharmacokinetics of the antibody in general, which may help with molecule design and making sure we're getting enough FcRn recycling, for example. Typically, these are measured by immunoassay. Reagents are generally easily sourced, so using target antigen and anti-human antibodies, we can set up a sandwich ELISA to measure these total antibody assays. But what we do need to do is consider the impact of the conjugated payload. And what I mean here is that if we have more or less payload conjugated, is that going to make the assay more or less sensitive to give you a response? And so what we can do is actually designing a characterization of the assay to assess the ADC and naked antibody admixtures to make sure that we are recovering what we think we're going to be recovering. Other end of the spectrum, we have the free payload. So the high potency of the payload means that we need to make sure that systemic exposure is kept as low as possible. These types of assays are also really useful in kind of early ex vivo stability studies where we're introducing the ADC into a matrix and making sure that linker chemistry is stable and is not leading to release of the payload. Generally, these are run by LCMS. And we can monitor metabolites, or we can look at attached linker with payload as well. What we do need to do is consider interconversion during extraction. So the ADC itself is likely to be present at a much higher concentration than the free payload. So we need to make sure that if we challenge the assay with ADC, we're not getting that interconversion into the free payload, which is going to interfere with our measurements. So then moving on to the conjugated assays. And on one side, we can measure conjugated antibody, typically by immunoassay. The reagent availability becomes a bit more of an issue here. And that's because we need to try and generate an anti-payload antibody to build up a sandwich ELISA. And we also need to consider the sensitivity to the amount of conjugated payload. So an increase in payload conjugation may not necessarily give you a linear increase in assay response. We can also measure conjugated payload, and this can be done with a hybrid immunoassay LCMS approach, whereby we pull down on the antibody and then we either cleave the linker and measure the free payload, or we can perform a protein digestion and measure some form of surrogate that has the payload, linker, and peptide attached. We do need to consider with this approach whether that linker is cleavable or not. If it is, that makes it a hell of a lot easier. We can use an enzyme to cleave the linker and just use the LCMS to measure the free payload. If it's non-cleavable, like I said, we'd need to use a protein digestion approach. But again, that kind of goes back to that conjugation technique and looking at the number of potential surrogates there may be for that particular analyte. And so if we consider a lysine conjugated antibody, that may be up to 80 different surrogates. That means it's just impractical to measure. Nevertheless, both of these measurements will measure your active form of drug product, but they'll likely not match one another. So the LCMS is likely to be sensitive to the number of payloads, whereas the immunoassay probably not fully sensitive. And we need to make sure we challenge the assays to characterize their conjugate sensitivity. Just wanted to briefly touch on drug to antibody ratio. This is a technique that is used to characterize ADCs after labeling to determine the average number of payloads on the molecule. But what we can actually do is implement this technique across an in vivo profile to see how that ratio changes upon dosing. And this can really help to contextualize those conjugate assay results. So it is a qualitative measurement, and we can implement that by using a high resolution mass spec approach. But because we're monitoring the molecule intact, it does offer limited sensitivity. So just bring this all back around. ADC measurement interpretation, I guess just to mirror what the other two speakers have said, a single assay measurement is not going to provide you with a fate of a heterogeneous drug product. So what we're going to have to do is utilize a combination of assay measurements, but also importantly, coupling that with a detailed understanding of those assay limitations to build an overall picture of the pharmacokinetics of the drug product. So just to summarize, complex medicines present complex bioanalytical challenges. So we do need to carefully consider what needs to be measured, and also what can be measured. And often we're looking at employing novel approaches to perform these measurements. If we are employing these novel approaches, we do need to look at characterization of those assay limitations to really truly understand that assay measurement. And it all kind of boils down to close communication between the bioanalytical and the project team. And with that, I'd just like to thank you for your attention. And there's a website address there if you'd like any more information on the drug development solution business at LGC. Thank you.
Thank you, Rob, for a really clear talk. I think we nearly reached the top of the hour, so I'm not going to ask you any questions on this occasion, but I hope that the audience found your talk as clear as I did. And I will just take this opportunity to thank all three speakers for your talk this afternoon. And we are heading towards the bank holiday weekend and half-term week for many of our audience, so we're skipping next week, but we will be back on Tuesday, the 8th of May, taking a glance at novel drug delivery systems. And we will be joined by Yvonne Perry from Strathclyde University, talking about lipid nanoparticles and telling us why they're so much more than a little fat blob. Then we will hear from Louise Coletta from the University of Leeds, who's bursting with promise, and she will tell us about precision drug delivery with therapeutic microbubbles. And finally, we will hear from Helen McCarthy, CEO of Fion Therapeutics, who's going under the radar with alternative delivery of mRNA vaccines. So I look forward to seeing you all in 2 weeks, and thanks again for our three speakers today and three really excellent talks. Thank you. Goodbye.